Professor CM Markan is an associate professor in the Department of Physics and Computer Science at the Dalbagh Educational Institute. He is a doctorate in physics from IIT Delhi. He has worked in the areas of solid-state physics, condensed matter physics, analog and digital integrated circuits, and VLSI design techniques. His research interests are analog adaptable VLSI, neuromorphic systems, and cognitive sciences. He has won several awards and honors and delivered invited talks at various seminars and conferences all over the country. So I would like to request Professor Markan to go ahead.
Thank you. Well, I take the cue from Professor Hemroff's work, where he's introduced about gap junction synchrony, and I would essentially be looking for a role of gap junction synchrony in developing or evolution of the brain function. And to make sure that we are all at the same level, I would be a little bit of introduction before I get down to the job. My talk essentially would be organized in the following points. Basically, I'll talk about the organization of brain function, genetic versus adaptive functionality, then neural development paradox, which we'll try to solve by means of role of gap junction synchrony, and then I'll try to talk about some of my work which I've done in terms of designing integrated circuits on the base of these brain function neurons. Well, we are all aware that mammals perceive a real world by 5 different senses, such as smell, taste, sound, touch, and vision. Perception of sense occurs because there is a dedicated area in the brain to process it. In absence of it, or any damage to it, the ability to perceive that particular sense is lost. So if you look at the brain, it basically consists of different areas, as you can see it here: the motor, the olfactory, the auditory, gustatory, somatosensory, and of course the visual, which is one of the most well-studied areas. And this is the same story whether it's for a cat or is it for the rat. I mean, basically all mammals have more or less the same structure. The organization of this particular sensory pathway essentially is in a manner that a sense of touch trickles down through action potentials right over the area in the brain which is responsive to this. In this particular case, the somatosensory cortex is responsive to this. The receptors in this particular case is touch, pain, heat, and other muscular receptors which are there. The story is more or less in similar ways in the auditory pathway, where the cochlea converts the frequencies and converts them into action potentials, which then travel over to the auditory cortex and make an impact there. In this case, the receptors are frequency, intensity, and localization. When we look at the visual pathway, we get almost the same story. From the eye, the rods and the cones convert the light into action potentials, which again travel through the LGN over to the visual cortex, which is at the back of the brain. And the receptors in this case, of course, are rods and cones. Another story that is to be understood is that there is a 1:1 mapping, or what is called a topological mapping, on the brain surface of the cortex with respect to different areas of the body from which the somatosensory cortex receives inputs. And the fact remains is that the nearby points on the sensory surface connect to nearby cells on the cortical surface. That's one of the principles that is important. And the second is the area occupied on the cortical surface reflects the sensitivity. So in that case, if you look at this particular case, the lips are occupying a larger area, so they must be more sensitive. If you look at maybe the arm, which has got a lesser area, is less sensitive, but your thumb is probably more sensitive, and so on and so forth. And you find that some of these areas are closely matched. For example, you would say the arm is closely matched to the face and also closely matched to the upper arm, and therefore any particular damage to your hand would actually be filled up by areas from nearby areas. Well, that's other point of the story. The same story sort of repeats in the case of hearing, and this is called the tonotopic mapping. And what you find is that in the auditory cortex, there is a proper organization in which the frequencies are debugged from an auditory signal that we have. So basically, you have a cochlea, which if you unwind, would have an organization in terms of the sensors organized in a particular manner to detect different frequencies. And then these therefore map onto the cortical surface as EE and EI, which stands for one ear and the other ear, and that some kind of alternate left ear, right ear type of mapping which is there. If we see the story which is there in case of vision, we would find that we have the we have this portion which is the visual cortex, which is getting inputs from this eye, and the mesh that you see here is the mesh that you see here, and therefore there is a topological mapping here. And if we were to open up the visual cortex, we would find that if this was the map in the real space, then the same thing mapped on the cortical space would be over here. This is the central part, which is largely represented as compared to the large outer part, which is less represented, and therefore the center of the eye is more sensitive as compared to the corner of the eye because it occupies larger area on the cortical surface. So that's the basic message that I wish to push across. So if you were to see an image on the screen, the way you will visualize is you will have more sensitivity at the center because it's got large mapping as compared to the peripheral parts that you see there. So how the different parts of the areas are mapped. Now let's look at this particular portion. Now these are the two eyes from which the inputs are going to the visual cortex, and this represents the magnified view of the visual cortex, in which the connections from the left eye, that is red, marked by red, and the connections from the right eye, that is marked by blue, are initially overlapping. So when the cortex gets formed, there is an overlapping of these connections. But over a period of time, when the activity starts, you find the segregation takes place. And that segregation is basically a competition between the left eye and the right eye, and they become mutually exclusive, such that one portion of the cortex is responsive to one eye, another portion of the cortex is responsive to other eye, which is essentially called the ocular dominance. So if I were to see it, if I were to open up the cortex and take a section of it, I would see, if I mark them as left eye and the right eye, I would see them as stripes. And if I were to see it in a larger portion of a cortex, I would see them as a very organized structure. Now that should make you feel as to who organizes the structure, because a huge portion, it's a very large portion, 5 mm, so it's got billions of neurons involved in it. And there's a huge amount of global order that is involved inside it. The second aspect that I would like to understand is what is called the orientation selectivity. That is, if I on the screen, if I show a particular oriented bar in a receptive field, and if I were to take an electrode and try to measure the activity in the visual cortex, then I find that when this bar is perfectly aligned to the receptive field, there's a heightened activity, but otherwise the activity weakens out. And this was the orientation selectivity of the cortical cells, which won David Hubel and Wiesel the Nobel Prize for this. So, so therefore, the way it is organized is, when the bar is right in the middle of the receptive field, the response is maximum, otherwise it is generally weak. And what Professor Hubel interpreted was that there are some on-center and off-surround cells which are aligned in a particular manner. So this is the cortical cell which is receiving inputs from the LGN, which is the intermediate part. So from the eye, it goes to the LGN and to the cortex. So there are intermediate parts which have got this center surround type of receptive field. So if you mix these center surround receptive fields, you have this on portion there surrounded with an off portion here. So if you look at this, when this bar comes right in the middle, this gives you the maximum response, and that is what they proposed as an orientation selective cells. Now all of this has to be created inside the brain. Now this was just one orientation selective cell, and therefore people came out with optical imaging. So this is a cartoon of that optical imaging, in which a monkey which is anesthetized, and he's able to see on the screen, and some activity is recorded on the cortex, which is then represented on this monitor. So when you have these grating type of bars oscillating, you find that certain portions of the cortex become active, the black ones are active and the white ones are inactive. So this is coded by this bar. So this is the vertical bars which are shown. Now when the bar turns around, you have a different activity on the same portion of the cortex. And therefore, if you repeat these gratings in, let's say, but they normally repeat with higher resolution, but in this particular cartoon I'm trying to show you with lesser resolution. But what you're trying to see is that now we're trying to look at the larger portion of the cortex, and they're able to see that there is a relationship between the different oriented cortex, oriented responses to different oriented bars. And when you put them all together, you see a very interesting result, which is of course intriguing the scientists as to how they go about doing it. So when we put all of them vectorially, and then color code them according to their orientation, we get a very fancy looking pattern which is here, and this is exactly what we're trying to see it here in this case. And when you see it in a larger scale, you see a very interesting pattern over here. Now that, if somebody is from the reaction diffusion background, would understand that these are patterns which can easily be created by reaction diffusion equations. And therefore, these patterns that you see are all color coded like this. They have got very important properties which are not, I'm not discussing those things here, but what I'm trying to raise is, an important thing is that when you look at the cortex and you see the response of the cortex, you see very interesting aspects. Now the question that comes up to our mind is this genetically programmed or is this adaptable? That's the major question. It's a huge amount of information. The ocular dominance maps, like the zebra stripes which you saw, and this previous pattern that you saw in this case for the orientation selectivity, and there are many others like direction selectivity, and there are many other things which are there. Are they pre-programmed or do they get adaptably developed? So let's try to address this question also. There is actually a timetable in which different aspects get developed. So you have this orientation selectivity developing here and orientation map developing here. By the way, this is the age, this is embryonic age of a ferret. Ferret is almost like a cat. And this is the postnatal age of the animal. And the animal is born right here, but it opens its eye somewhere down here. Right? So there are some things happening before it is born. There are some things which happen after it is born. And there are other things which happen before the eye opening. And there are other things, of course, happen after the eye opening. Just now what I have just shown you is all the things that are observed after the eye opens. But the fact remains is that the orientation selectivity develops even before the eye opens. So that means there is a part of genetic programmability inside it. But how it can be done is the question where gap junction synchrony would have to put an answer. Right, so that's the basic idea. So there's a timetable and we find certain things develop even before the eye open or even when the input has, or the visual input has not even reached the visual cortex. Rest of the things, of course, develop according to the visual cortex. Let's look at a very simple thing. This is the ocular dominance stripes, the left eye and the right eye balance, when both the eyes are equally active. And this is the pattern which occurs when one of the eye is sutured or closed down at the point of birth. So there's no visual activity has gone from this left eye, all the activity has gone from the right eye. And because this was developing according to the competition is lost towards the right eye, and therefore you find it's more white than black. So therefore it is adaptive. So if you change the activity, the brain or the cortical responses do change. Now this is a summary slide which tells you that if you have, let's say, from the left eye a connection going to the cortex, to both the portions of the cortex, and this is the right eye connection going to the cortex. So now if I activate the left eye, I activate this portion but not this portion, for example, and from the right eye this cell and not this cell. So we find that there's a mutual exclusiveness of activity between the two cells. Although they are mapping to both the cells, their segregation will take place, and this portion will become left eye dominated and this will become right eye dominated, for example, like a black and white stripe in this case. Now this happens because of the Hebbian rule that says cells that fire together wire together. So we had these four cells. These two fire together and this also fire together, so they both wired together. But these cells and this cell, or for example this cell and this cell never fire together, so they become mutually exclusive. This cell and this cell never fire together, so they become mutually exclusive. So the rule is the cells that fire together wire together. Or you could even say that cells that do not fire together compete for elimination. And that's alternate way of looking at it. Well, the question is this the story only for visual cortex, or do other cortices also follow the same rule? A very important experiment that was done by Professor Mrigangasur from MIT, and what they did was, they said, let's say there are two types of inputs coming. One input is coming from the eye and going to the visual cortex. Another input is coming from the ear and going to the auditory cortex. So they are exclusive paths. What they did was, they rewired the structure. That is, they took visual input and through a particular relay station, send the visual input to the auditory cortex. So now the auditory cortex is receiving the visual input as well as the visual cortex receiving the visual input. And what they found was that the orientation maps which normally develop in an animal also developed inside the auditory cortex. Auditory cortex, if it had been genetically different, then would not have developed these visual maps. And the second thing is that when you show any light corresponding to this area which is going to the auditory cortex, you begin to hear something. So the cortex understands auditory cortex in that context because of the placement of that. But here there's a rewiring there, and therefore when you are shown to the eye, you begin to hear something, and that is trying to tell you that different cortices are not different, but they are different because they receive different inputs either from the ear or from the eye or whatever that may be. So similar mechanisms operate in generation of receptive field properties in different cortices. Cortices are similar at creation but differentiate depending on the sensory modality they process. Prediction from one cortex can easily be extended to the others, and non-performing sensory cortices may be taken over by other active senses. So for example, you're born blind, you will have an acute sense of hearing, essentially because the wires will start going towards that part, and that will have a larger representation on the cortex, because the visual cortex will also start responding to auditory cortex, and therefore the auditory cortex, which was supposed to have a smaller area, will begin to have a larger area, and therefore higher sensitivity will develop, and therefore people who are born blind have more acute to their senses. This of course was done by Mr. Megan Kasoor, who also mentored my work during my PhD work at IIT. So the question is, so much of different sensory cortices, so much of different representation, how do we begin to see? So the problem is what is called the binding problem. Every time you see an object, the brain processes the image by splitting it into different components. We saw at least 2 or 3 components, but every sense splits it up. But we perceive the object as a whole. This is what is called the binding problem. What we need to do is stitch information in the form of objects. The neurons that map the features constituting a scene are stimulated, and they become vigorously active, emitting rapid bursts of action potentials, which are called firing of action potentials. These collective action potentials in a neighborhood are recorded in the form of neural oscillations, also known as EEG. These EEGs, or what are called neural oscillations, are fundamental mechanisms enabling coordinated activity during brain functioning, and they establish temporal correlations between distributed neuronal responses, and heightened synchronized neural activity segregates the object's features in a scene. So that's generally what binding problem is solved. The synchronized oscillatory activity of different neurons, when they get coordinated, they stitch up into one particular coherent object. And on the basis of which there are different types of EEG waves are classified. They're called gamma, beta, alpha, theta, and delta waves, and they have their different tasks. So we come to what is called the neural developmental paradox, and that says what we require, 2 things we require, is that we require this feed-forward connectivity, that is receptive fields, to develop, and the second thing we require is synchronization between 2 far-away neurons. Because if I want to talk about an apple, I begin to smell an apple, I begin to see the spelling of an apple, and they are all represented in different cortices, but they must stand together as one coherent object, right? So therefore, these are very distant neurons, and the only way to make them fire together or get synchronized, there should be a link between them, and that should be through an axon. So the question is, does this link develop first, or this vertical connections develop first? Because if this link were developed first, then that means you have a genetic information to know that this cell is going to look at the picture of an apple, and this neuron cell will look at the spelling of an apple, which, as we said, everything is adaptive. You can't really say which portion of the neuron will be responding to what. And therefore, how can this connection ever be there? And if this connection is not there, how do we synchronize the activity of these 2? So because unless the 2 activities, or let's say these cells fire together, they will never wire together. So that's a bigger problem, that unless these 2 cells fire together, they will never wire together. So who brings this, the long range connection into the play? So that's the major problem that we would like to address. And that's where I think the gap junction synchrony plays an important role. So what people propose is there are 2 phases in which the cortex develops. In the first phase, the mechanisms inherent to the cortex establish a crude framework on the basis of genetic information of interconnected neural modules which exhibit basic but still immature traits of adult state. And in the second phase, the primordial layout becomes refined under the control of visual inputs or visual experience, establishing a fine-tuned network of connections and mature response properties. So therefore, the current emphasis on correlation-based models, which may be appropriate for plastic changes, could be obscuring the role of intrinsic signals that guide the initial establishment of the functional architecture. And therefore, there is always a doubt now is whether the visual activity alone, based on the Hebbian principle, is sufficient to make these maps develop. So the question is, what mechanisms would mediate the phase 1 of the cortical interaction as to who coordinates the correlated activities of the 2 distant neurons? It has been suggested, and Professor Hameroff is one of the original authors for these kind of things, is that say besides chemical synapses, neurons also interact through direct electrotonic connections with their peers via electrical synapses. These electrical synapses are what are called gap junctions. That is where you see it here. These are like unofficial connections. They are not formal connections. All formal connections are the chemical-based synapse, and these are actually very slow. Electrical synapses or gap junction mediated connections are reciprocal pathways for ionic currents and small organic molecules. And they, as Professor Hameroff has well been suggesting, that they are often strong enough to mediate close synchronization of subthreshold and spiking activity among cluster of neurons as far away as almost across the brain. So you don't require a physical connection. You can actually have a quantum entanglement through microtubules from one portion of the brain to another portion of the brain and can possibly synchronize it. Electrical synapses have recently assumed importance as quantum entanglement of microtubules in the dendritic network has been suggested to occur through gap junctions generating collective neural oscillatory activity in the gamma band of 30 to 80 Hertz, attributed as possible neural correlate of consciousness. And we thank Professor Hameroff and Professor Penrose from bringing this into focus. But the fact remains is, biologically speaking, the expression of neuronal gap junction is highest in early development when synapses have not yet developed. So these neural gap junctions are in abundance when let's say an animal is born or a child is born. But they progressively decrease as synaptic chemical activity patterns are established. So they are precursors to chemical activities that occurs. But it's not reduced to zero even in mature and healthy networks, and that is why the consciousness concept of using electrical synapses is being studied, because they are also occurring in adults as well. But they have an important role in the development part. It appears that the gap junctions do not play a purely pathological role in shaping the network dynamics. They also probably play a role in consciousness, which Professor Hameroff has been talking about. In fact, 44 to 100 Hertz gamma brain oscillations increase while participants are studying words that they will successfully as opposed to unsuccessfully recall. So the heightened activity is there when they are in the formation of or hardwiring the connections between distant neurons. Gamma activity is reactivated just prior to recalling an item, with higher levels of gamma predicting whether or not the recalled items are actually studied. Gamma rhythms have also been found that they can act as preprocessors for beta rhythm, allowing networks displaying a beta rhythm to maintain cell assemblies. Cell assemblies are distant connectivities which are all representing the same object established during gamma rhythm. Gamma and beta rhythms may play complementary roles in processing, with the gamma rhythm preparing the beta rhythm for participation in long distance coordination. So the functional significance in the brain development and the interrelated chemical electrotonic transmission at the mixed synapses is an important question, and we feel that the role of gap junction mediated synchronization of neural assemblies as a possible source of rapid, informal back-channel communication promises to break the neural developmental paradox as to who creates the synchronization. Is it a hardwired wire, or is it something other than that? So what, if you try to see the same diagram in a cartoon form, we find that this axonal lateral connection could come later on. Initially, there's a coordinated activity which can occur through dendritic interaction, through gap junction mediated synchrony, or through what is called quantum entanglement of these faraway microtubules, so that both the neurons are active at the same point of time. And if these neurons become active at the same time, then Hebbian rules will ensure that those cells which fire together will begin to wire together, and then this connection would be there. So when you see an apple, you will begin to see the spelling of an apple automatically, because then it becomes hardwired. So when you remember multiplication tables, you put all your attention to understand that, but later on all your mathematics simply flows out of you, because then it all gets hardwired. So the hardwiring takes place much later, and that's what I talk about, the brain function has to be taken there. So role of gap junction mediated synchronization of neural ensembles as a possible source of rapid back-channel communication promises to break the neural development paradox. So broadly, the importance of synchrony essentially is not only to bind features, to provide selective attention that helps to focus attention on one neural assembly among many in the background. That's where consciousness is normally addressed. Not only that, it also provides a time window that ensures, or rather, when I mean time window, I mean synchronization, to provide that ensures that some recurring neural assemblies may be hardwired, which Professor Christoph Koch calls as zombie modes, or for example, when we learn cycling, we pull our attention to it, but later on we cycle without all our attention to it. This is essentially called a zombie mode, where it gets hardwired into us that how to balance is an automatic phenomena. We actually addressed this problem avoiding gap junction synchrony at much earlier date. So if you look at this particular, it doesn't come here. So in a paper in 1999, we actually proposed that the competitive learning could actually take place because of a leaky chemical that will be coming out from a neuron, thereby sending a chemical message between one neuron and another neuron. And that at that particular point of time was proposed to be a nitric acid which comes out from an active neuron, and that it helps the nearby neuron to get activated. So we use this as a model. So instead of being isolated activity, we introduce what is called the diffusive chemicals coming out of an active neuron to try to create a Hebbian learning, which we define as diffusive Hebbian learning as an isolated competitive Hebbian learning. And what we found out, that when you introduce this leaky chemical or introduce in modeling this leaky chemical coming out from an active neuron to the nearby neurons, we could actually create a mechanism in which we can say, in a similar neighborhood, the biases are strengthened of two neurons in a similar neighborhood are with a bias strengthened. In the opposite neighborhood, the biases are reversed. But if they're a mixed neighborhood, the biases become unbiased, and therefore you could actually create some kind of a gradual variation of function over the cortical surface. Lately, what we have done is we've developed an integrated circuit or using VLSI technology to be able to create on-chip analog memories. So these analog memories are controlled by quantum tunneling and injection, and they can actually be modified, and they're non-volatile. So in simple words, they are like analog EEPROMs, electrically erasable programmable read-only memories, which are digital, but here it is actually analog, and therefore you can control weights of the neurons at a very fine precision. So we have this floating gate and another floating gate, and we have these two parallel circuits which are modeling the input from the left eye and the input from the right eye. So it's a cartoon. In some cartoon it looks like this. So left eye and the right eye, and a cortical cell is getting connection from both of them. When the competition takes place, there's a segregation that takes place, and whichever arm is more stimulated or has got a small bias as compared to the other, one of them wins and the other one loses, and finally you will have a competitive state as either this is connected to this or this is connected to this. So if this is the left eye and this is the right eye, so we'll say this is a right dominated cell and this is a left dominated cell. So we could actually do it in an electronic circuit, all this actually trying to happen here. Not only that, we actually took this cell and put it on a two-dimensional RC network, and we could show that over a large area we could create stripes like ocular dominance stripes, and all of them was done in 0.35 micron CMOS technology. And they can actually be modeled by simple reaction diffusion equations, which are normally called the Turing patterns, and we could easily create either mathematically or through these electronic circuits. These can easily be created only because we had this diffusive chemical coming out from a neuron, or some kind of a chemical linkage was taking place. We extended our job a little more, and we said, can we really create orientation selectivity in an integrated circuit? And therefore we tried to wire up these competitive networks in a vertical form with, this is an on-center and this is an off-center type of cells which are being given to this, and then so therefore this is what we wanted to emulate. We wanted to create orientation selectivity. The on-center cells should all get aligned and off-center cells should all get aligned. So this is what we began with, and what we did was we started with completely unbiased situation where cells had random biases. So some of the cells were actually biased towards on-center, and some of them tried to bias with off-surround. And later on, when the competitive structure took place, we found that we had on cells in the center and off cells at the surround, and therefore you can say that this cell is actually orientation selective, because if I show a bar parallel to this, this will highly respond as compared to that, and that has also been shown here. So if I show an input aligned in this direction, you get a maximum response, otherwise you get a minimum response. So we were able to create these things in electronic circuits essentially because we had this leaky chemical with no idea as to whether we can actually exploit the gap junction synchrony. Professor Hammeroff himself has done a work which has been recently published in 2011, in which they have also tried to model with Mark Ebner, in which they try to model a neuron in this complicated structure, which is called an integrate fire type of neuronal structure. And they also take this resistive grid on which this cell is connected, and they're able to show that if you look at an image of, let's say, this flower, and there are several cortical cells looking at this flower at different directions. Then on this network, they can have what is called the flow of conscious experience moving around from here, and they are autonomously moving. So it is able to, segregate this flower from the background almost on its own, and therefore sometimes the flower will get activated and sometimes the surround will get activated, and therefore you have an asynchronous response of the flower versus surround, and therefore by synchrony you are able to segregate the flower from the background by simple gap junction synchrony that is taking place. So what we hypothesize is that initially the coarse assemblies are recruited as they increasingly get hardwired. The synchrony builds assemblies that are both more refined locally and well connected globally. This adaptive mechanism of building neural assemblies helps customize zoom beam modes for every individual. The gamma synchrony reaches its maximum at the adulthood by the time most of the essential zoom beam modes are in place. We thank the author is deeply beholden to Most Revered Chairman Advisory Committee on Education in Diyarbakir for being the beacon of motivation and inner strength and inspiring this study. Thank you.
Do we have any questions?
You linked brain with conscious experience.
That's right.
And the activity of cortices, you said that whether it is genetic or adaptable.
That's right.
So do you think there is some, what about the impressions of previous births that we call samskaras? Do they also play some role in these things?
Well, we all understand and generally appreciate this part that evolution takes place. There is a genetic refinement that takes place. So as a body, we share a lot of information from our parents, and therefore genetically we are made in a particular manner, and some of these connections will build according to that. So if you stop using this hand, let's say full 10 generations do not use this hand, surely the capability of using this hand will be lost. So we have been over a period of time being inculcating or generating certain sort of habits which get hardwired in ourselves, but there are many other habits which we don't use get lost. And therefore there is when I said before eye opening, there is always a network which is there, but that can be lost if you use different activity altogether. But if the visual activity reinforces what has been encoded, it will become or resonate or will become stronger. So if you are talking about the rebirth essentially is through the evolution and not otherwise. I mean, that's what I understand.
In the early part of your talk, you mentioned an experiment, if I understood correctly, that the visual input was directed into the auditory part of the brain.
That's right.
And the subject heard sounds.
That's right.
So were these done on humans?
Well, no, it's ferrets. Ferrets is a family of cats.
So how do they know that the animal heard the sound?
Well, they train these animals. They psychologically train these animals. So when they hear a sound, they pick up, let's say, a red bottle, or they kick the red bottle, or if they they see something, they kick the blue bottle. I mean, there are ways by which they control animal behavior, or they are able to observe the animal behavior.
Were they repeated at some other lab?
Yes, this is a very, very famous experiment which they have been able to do this part, and it's been repeated at many other places.
Okay, thank you.